Hello, welcome to Physiology with Shakthi, and today we're going to do with insulin signaling pathways. So, this is one topic I think as postgraduates you would have come across in various different things. When you talk about aging, when you talk about the normal insulin signaling pathway in reference to cancer. So, ultimately, this is kind of the future that we're looking at. This one pathway of insulin signaling. A lot of things happen, and it explains your metabolic as well as the mitogenic properties of insulin and insulin like growth factor. So the first thing is the receptor. When you talk about the insulin or insulin like growth factor, you know that it has an intrinsic tyrosine kinase activity, and this receptor has a alpha and beta, and it has 2 alpha 2 beta. So when we talk about the structure, this is the membrane. So if this is the insulin receptor, it has an alpha and a beta domain. The alpha domain is extracellular, beta domain is intracellular. So this, when a ligand like the insulin or the insulin like growth factor comes to this receptor, there is activation of that receptor, and there's something called dimerization of the complex. So because of that, what happens is there will be activation of the intrinsic tyrosine kinase activity. So the ligand binds, there is activation of this receptor. So basically what happens at that time is your intrinsic tyrosine kinase activity is activated. So that dimerization leads to activation of the intrinsic tyrosine kinase activity. And tyrosine kinase activity, what does it mean? Into tyrosine residues you phosphorylate. So this is going to phosphorylate the other segment, and this is going to phosphorylate the tyrosine on the other segment. So ultimately that will make both of them active. So this receptor is now active. So because of the ligand binding, that result in dimerization and intrinsic tyrosine kinase activity, resulting in activation of the receptor. So that is not only the things that this results in phosphorylating. There is a group of usually a cytoplasmic protein. So this cytoplasmic protein is present somewhere here like this, and the cytoplasmic protein in this case is generally given a term IRS, the insulin receptor substrate. So this is the substrate on which it acts. So this IRS is the cytoplasmic protein that we are talking about, and in that there is a tyrosine segment. So these tyrosine segments in the cytoplasmic protein gets phosphorylated now. So this is now phosphorylated. So not only does it activate itself, some associated cytoplasmic proteins are also phosphorylated. The tyrosine segment in them are phosphorylated. So what does that do? It results in completely different effects. So we'll start off with one thing. This is insulin binding. When insulin binds, what should happen? What does it ultimately lead to? An insulin mediated GLUT4 transporter is given to the surface so that glucose can come in, and that glucose comes in, the cell should start utilizing that glucose. Glycolysis should take place because of the glucose, and excess amount of glucose should be converted to glycogen, or it should be converted to the lipid. So lipogenesis or glycogen synthesis, and also things like gluconeogenesis. We don't need that now. We have glucose and we need to use glucose as the energy store. So we don't need gluconeogenesis, and that should be inhibited. So these basic metabolic things should happen because of this. How does that take place is what we're going to find out right now. So when we look at this has been phosphorylated and this complex is active now. When this complex is active, there is another set of proteins that are present. So this is a P85 molecule here. There is a P85 molecule over there, and attached to that P85 molecule is a Pi3K molecule. So there is a Pi3K molecule. This P85 normally inhibits this and keeps it in place. What this is, I'll explain. Wait. So this P85 kind of inhibits Pi3K and keeps it over there. So phosphorylation of your IRS, the tyrosine residue there, that is now going to free this. So the action of the inhibition of Pi3K by P85 is no longer there. So that gets freed. So your Pi3K is now free to act on something. What does it act on? So when we look at the membrane, your membrane has things like the PIP2. So if you attended my class on signal transduction, the G protein coupled receptors, if you haven't, take a look over here. I'll put a suggested link over there. So if you attended, you would have been knowing by now that it is phosphatidyl inositol biphosphate. So phosphatidyl inositol biphosphate. So two phosphates are there. That phosphate is there in the 4th and the 5th position. What Pi3K does it is phosphatidyl inositide. So it is phosphatidyl inositide 3 kinase. So we told in the 4th and 5th position there is phosphate, that is biphosphate. Now in the 3rd position, one more phosphate is going to get added. So this results in phosphorylation in the 3rd segment. So 3, 4, 5 triphosphatidylate. It is going to become a triphosphatidylate complex. So this will be now a PIP3. So in the 3, in the 4, in the 5 segment, you got the phosphate groups present now. Understood what that does? So that is Pi3K, phosphatidyl inositide 3 kinase. Understood? So once that is active and once this is now present, this acts as an attractant for certain set of molecules. So one set of molecule is your PDK. This is your PDK1. So that is one set of molecule that is going to get attracted over here. So what does PDK1 means? So that is 3-phosphatidyl inositide dependent kinase. So it is a 3-phosphatidyl inositide dependent protein kinase. So when this is activated, based on that, now this gets activated and this is a kinase. So what does that do? That is what you need to understand. Another group of molecule is also going to get attracted over here. That is your PKB AKP. So this is another molecule that is also going to get attracted over here. Are you with me till now? Yes. So this PKB is protein kinase B. We know protein kinase A in G protein coupled receptors. Alpha S, that will stimulate the protein kinase A ultimately. Yes. And we know the protein kinase C in phospholipase C activated pathway. The DAG will finally activate protein kinase C. So like that, it is protein kinase B. It's also called AKT. So when this was discovered back in 1991, 3 different group independently discovered this. So it was kind of related to the pathway where the transforming leukemia virus AKT8 was involved. So that is why it's called AKT. So we got the protein kinase B or the AKT. Yes. Now, this protein kinase B or the AKT has two residues. One, a threonine residue, and another serine residue. So this is another important concept that you should understand. When we talk about intracellular signaling, tyrosine, serine, threonine. These are three important amino acids which play important role in intracellular signaling. There are tyrosine kinase associated. There is serine threonine kinase. So apart from that, there are phosphatase also. But for now, these three are very important in intracellular signaling pathways. So this 3-phosphatidylate dependent protein kinase, what that does is it is now going to phosphorylate the threonine part of your PKB. So that will result in some sort of a partial activation. So that is a partial activation. You got me? Yes. So along with this, just keep in mind there is another thing called the mTOR. Have you heard of mTOR before? Mammalian target of rapamycin. So if any UGs are watching this, that is an MCQ actually. Rapamycin, so anti-aging drug. But because of its side effects, you don't actually use it as an anti-aging drug. But that is how it kind of acts. This molecule over here is targeted by the rapamycin. So that is why it's called mammalian target of rapamycin. So what this does is it is now going to phosphorylate this, and that will result in complete activation. So how does a partial activation result in this complete activation? So you need to understand that this mTOR is normally inhibited by a tuberous sclerosis complex. A tuberous sclerosis complex. So there is 1 and 2. Two complexes are there. 1 and 2. Two separate things are there. So these tuberous sclerosis complex normally inhibits this. The mTOR is inhibited by tuberous sclerosis complex. So when the protein kinase B is activated, it results in phosphorylation of many things. One among them is this. So phosphorylating this kind of inhibits this activity. So inhibition of this inhibition means now your mTOR is free to go and phosphorylate the serine residue. So that results in a complete activation. So you see what is happening here? Initially through this, through PDK1, it resulted activation, which is a partial activation, and then that is followed by phosphorylation of tuberous sclerosis complex, which is now going to inhibit its activity of inhibition of mTOR, making it available to now phosphorylate your serine residue. So complete activation of protein kinase B happens. And what does protein kinase B finally do apart from this? Protein kinase B goes and acts on a molecule, a protein molecule. So this molecule is a 160 kilodalton molecule. So that is why it's called AS160. This is a molecule which is AS160. So AS160, once it's activated, will result in translocation of your GLUT4 receptors on the cell membrane. So these GLUT4 receptors will now go onto the cell membrane. So that is one of the things that happens. Now GLUT4 receptor has gone to the cell membrane, glucose is coming in. Now glucose becomes glucose 6-phosphate, that becomes fructose 6-phosphate, and that should become fructose 1,6 biphosphate, and it should commit itself to glycolysis for the glucose to be utilized. Am I right? Yes. So For that, your phosphofructokinase should be activated. So that activation of your phosphofructokinase is again through phosphorylation of your PKB. So your PKB phosphorylase phosphofructokinase, and that is now going to result in your glycolysis. Now glycolysis is taking place, all fine. Yes, but when there is excess amount of glucose, what should happen to that glucose? It should get ultimately converted to either fat and stored or glycogen and stored. So that should happen. So normally you have your glycogen synthase. So that glycogen synthase is inhibited by a complex called glycogen synthase kinase. So your glycogen synthase kinase kind of inhibits your glycogen synthesis. So now this PKB is again going to go and phosphorylate your glycogen synthetase kinase. So when this inhibition is now lost, that will result in glycogen synthesis now. Also, this also stimulates your lipogenesis. So GLUT4 transport, glucose is coming in, committed to glycolysis, excess glucose through this will result in glycogen synthesis and lipogenesis. So these are few metabolic things that are happening. Apart from this, you know that gluconeogenesis takes place when glucose is not there. Yes, so you need to inhibit or suppress your gluconeogenesis in the liver. So we need to suppress gluconeogenesis in the liver. So in order to do that, one thing that happens is through your PDK1 directly, and another thing through your PKB as well. So if you think about it, when gluconeogenesis needs to be stimulated through catecholamine action, that is a beta mediated action. Beta is alpha stimulatory. Alpha stimulatory adenyl cyclase cyclic AMP that will bind to CREB, cyclic AMP response element. That is a transcription factor, right? So this CREB is a transcription factor which results in. Now what this does is that is now going to disassemble this. So your CREB complex is disassembled. So disassembly of the CREB would result in suppression of gluconeogenesis. Along with that, there is another transcription factor which is called FOXO. So this will phosphorylate FOXO, and that is going to act in suppression of your gluconeogenesis. So this is fork head box O, that is FOXO. So fork head box O, that is your FOXO. So that is also now going to inhibit your gluconeogenesis. So these over here are the metabolic effects. So I'll draw a line over here. These are set of metabolic effects because of insulin cascade. The signaling resulted in these metabolic effects. So here I'll help you understand how the mitogenic action happens. Mitogenic in the sense when the insulin growth factor, so insulin like growth factor, insulin, all of them seem to have a proliferative mitogenic action. How is that taking place? So before that, there is also another pathway which has both metabolic as well as mitogenic action. So that is where your mTOR is coming into play. So mTOR is going to activate your SRBEP. So this is the SREBP. The S stands for sterol, and this is the regulatory element binding protein. So the sterol regulatory element binding protein. So because it is sterol, it is going to result in your lipogenesis. So your lipogenesis is stimulated and lipolysis is inhibited. Is that clear? So through all this pathway, the metabolic effects have come into view. So if you take a look at it like this, it will seem very confusing, but if you had followed me step by step, it was actually fairly simple. So there was your phosphorylation of tyrosine residue of IRS that will remove the inhibition of PI3K, and that is now going to phosphorylate PIP2 to form PIP3. Now PIP3 acts as a attractant for molecules like PDK and PKB also. So finally this PDK1 is going to phosphorylate the threonine part of PKB. A partial activation happens, and through that it is going to inhibit the TSC, tuberous sclerosis complex, which normally inhibits mTOR. So now that inhibition is gone, mTOR is going to phosphorylate your serine complex. So this serine residue is phosphorylated, that is in complete activation. So what is it going to do? It is going to act on AS160 molecule, and the translocation of GLUT4 is present. So that will result in glucose to come in. Glucose comes in through activation of phosphofructokinase, glycolysis is committed, and through phosphorylation of glycogen synthetase kinase. Now it finally ends up relieving the inhibition, and stimulation of glycogen synthesis happens as well as lipogenesis happens. Also through PKB activating the FOXO as well as PDK1 disassembly of CREB, ultimately gluconeogenesis is suppressed. That's simple. And through mTOR, through the SRABP, it is going to stimulate the lipogenesis. So these are all the things that happen because of insulin over here. So now that we're done with the metabolic functions of insulin signaling, we'll go ahead to what happens and why it is mitogenic in nature. So this IRS with the tyrosine residue phosphorylated, so this actually acts as a PY motif, what is called a PY motif. So it is a phosphotyrosine domain. The phosphotyrosine, the tyrosine there is phosphorylated. So these are generally called what is the PY motif. Same thing. So these PY motif acts as an attractant for certain set of molecules. So in that, when we look into it, especially with regard to how growth factors and all act, you'll notice that there is a group of SRC family proteins. So this SRC family proteins can combine with, attract and combine with this PI motif that are present. So to the PY motif, certain things bind. So over here, example is GRB2. So this GRB2 is growth factor receptor bound protein. So these are growth factor receptor bound protein. And now this GRB2 is going to attach through a SH part. So this SH stands for SRC homology. So over here SH2, the SRC homology 2 part. So I had already mentioned how PY motifs attract this SRC family groups. So that SH group attached to this PY motif, and this GRB is now attached. Once GRB is attached, it is now going to recruit a molecule called the SOS. This SOS gets recruited. So what is the full form of SOS? It is called son of semenless. I don't know why it's called so. If anybody of you knows, I tried finding it out. If anybody of you knows, please drop it in the comment. It will be helpful for me also. So that is called son of sevenless. So the son of sevenless is a guanine nucleotide exchanger. Are you understanding what is going to happen next because of that? It is a guanine nucleotide exchanger. If you still haven't figured out, let's think about our G protein coupled receptors. When we talk about the G protein coupled receptors, we have large G protein coupled receptors and small G protein coupled receptors. These large G protein coupled receptors are what you naturally see with the alpha, beta, gamma domains, and that alpha is linked to GDP. That is inactive. When alpha is linked to GTP, it becomes activated. In a similar fashion, there are small G protein coupled receptors. So these example of this is RAS. So in this RAS, there is no alpha beta big chains like we see in the large molecules. So this is a small G protein coupled receptor, and that is attached to a GDP. Now this son of sevenless is a guanine nucleotide exchanger. So what it does is it exchanges the GDP, which is attached to RAS, and makes it into a GTP, activating your RAS. So over here we have your RAS complex, and RAS is attached to a GDP. Now that gets activated and forms the activated RAS attached to GTP, and that is mediated by your SOS. Once that happens, this RAS is now going to activate your RAF. Now what this RAF is going to do is it is going to activate something called MAPK. So MAPK. So it means mitogen activated protein kinase. It's also called MEK1. So there is a molecule called MAPK, which is mitogen activated protein kinase. So this mitogen activated protein kinase is MAPKK. So once that is activated, that is going to activate your MAPK. What does MAPK do? It goes into your nucleus, and it goes into your nucleus, binds to a transcription factor, and that is going to result in activation of certain set of response elements and genes. So that will result in protein synthesis, and that is your mitogenic action. So that side we have seen the metabolic function. Here you can see the mitogenic action. So this MAP kinase is also called the ERK. So this ERK is called extracellular signal regulated kinase. So an extracellular signal regulated kinase. It's also called MAPK. Now apart from this, there are a lot of cross connections between them. You should also know that RAS is also going to activate your PI3K directly without acting on this. So without acting on P85, it directly acts on PI3K, mitogen activated protein kinase. So that will result in final mitogenic action. Now apart from this, there is also your mTOR. The mTOR can activate set of proteins like P70 and 4EBPS. So that is also activated, and through that again protein synthesis happens. Also I mentioned this is also helping in mitogenic action. So ultimately there is mitogenic action as well as metabolic action. So what is the importance of having to understand this signaling pathway? One is the insulin. This is a major insulin signaling pathway. So anything that changes, affects, blocks all this can be a potential therapeutic agent we can use. Yes, so that is one reason. And another reason is this is intricately involved in a lot of things. So when we talk about aging, insulin signaling pathway, aging are very correlated. A lot of studies have prove that. So, for example, age1 gene, knockout of that will affect PA3K. There is a clotho gene that affects the insulin signaling pathway. There is a prop1 gene as well. So all these, you take a look at growth hormone receptor knockout models, that is also going to affect. So ultimately all of them seem to live longer without this. So that is one thing. Your mTOR, I had mentioned earlier, mTOR rapamycin, that somehow results in blocking aging. And also there is the certain models and all that. Also when we talk about the metformin, for example, that acts on the AMPA as well as it has blockage of mTOR as well. So this, you can see how intricately linked it is to your aging. So that is one thing. Another thing is your cancer, aging, prolonging life, that is kept in balance, right? So life prolonged means high instead of cancer. Otherwise, there's aging. So it's kind of like a balance that is kept. So all these pathways are also important when we talk about cellular proliferation and in cancer. So how growth factors act. Excess action of this will result in excess proliferation. So this is very important when you talk about cancer, insulin pathway, aging. So this is going to come in a lot of things, and this is kind of like the future that we're looking at. So where a lot of effective therapeutic models can be developed from this. So that is it about this. So if you take a look at it completely together, it's going to be very confusing. You break it down into small components like I had done, then it's going to be very easy. Alright, thank you.